I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, excited to bring you episode number 73 in this podcast dedicated to the improvement of your own brain by any and all means at your disposal. We're going to be talking with a couple of guys today, colleagues from the Swinburne University of Technology in Australia, which is gaining quite a list of Smart Drug Smarts alumni. This episode actually spawned out of a conversation from episode number 71 with Professor Andrew Scully. He mentioned a couple of his colleagues who were studying the actual mechanism of action behind the benefits that omega-3 fatty acid, sometimes better known as fish oil, although that's not the only source, can provide in the brain. And I followed up on that, and today I'm going to be talking with Professor David Crewther and Dr. Andrew Pipingas all about omega-3s and omega-6s, diet, scientific study methodologies, and how much omega-3 supplementation might really make sense. If you hang around until the very end of the episode, I'm going to talk about something weird, which is evidence of a secondary, non-conscious visual system that humans seem to have. Almost a little bit sounds like weird psychic phenomena when you say it that way, which it's not. But humans seem to have an uncanny ability to understand their visual environment without doing what we typically think of as looking at it. So more on that in the Ruthless Listener Retention Gimmick. But first, let's kick things off with This Week in Neuroscience.
Smart Drug Smarts. This Week in Neuroscience.
So good news for anybody who wants to have full control over your sleeping and waking cycles. Scientists are making progress in this direction. There's been some research at Vanderbilt University that apparently recently discovered a reset button for the part of our brain that keeps track of our circadian rhythms. Circadian rhythms, sort of when you're awake, when you're asleep, when you're hungry, all these things that happen on a daily basis. They did this study using mice, as is so often the case. Mice apparently have a very similar circadian rhythm to humans, except it's just flipped on its head. We're diurnal and like to move around during the day. They're nocturnal and like to move around at night. But other than that, pretty similar. So there's a part of the brain called the suprachiasmatic nucleus that's inside the hypothalamus, and that's basically sort of the control mechanism for our circadian rhythms. Prior to this study, it had been the established wisdom that this is sort of a read-only device. It gets some inputs from the outside world as far as things like temperature and the type of light that an organism is exposed to. But other than that, the rest of the brain seems to be reading its signal, not really writing to its signal, and there wasn't a way of stimulating it directly. However, in this study, they used something called optogenetics to manipulate the firing rate of these suprachiasmatic nucleus neurons with light. Now, of course, it's not a normal thing for a brain cell to be responsive to light, but this is one of these crazy genetic experiments that you hear so much about, where they genetically engineered these mice to have photosensitive neurons within their brain, and in this case, within their suprachiasmatic nucleus, says Douglas McMahon, director of this study. We found we can change an animal's sleep-wake rhythms by artificially stimulating the neurons in the master biological clock using a laser and an optical fiber. They used 2 strains of genetically engineered mice in this study. The neurons in the brain of one strain contained an optically sensitive protein that triggered neuronal activity when exposed to light, and the other strain of mice had just the opposite effect. They had a similar protein that suppressed activity when exposed to light. And in both cases, they were able to essentially reset the circadian rhythm clock within the mice using this light-based on-off switch. Of course, this exact approach isn't ready for human use yet, said doctoral student Michael Tackenberg, but others are making progress toward eventually using optogenetics as therapy. And for those of us that already have our brains fully developed, we're no longer embryonic, and we are not good candidates for organism-level genetic manipulation, there's still a potential possibility of introducing new genes into living cells using viruses and things like that. So lots of super sci-fi possibilities here. And who wouldn't like to have full control of your sleep-wake cycles when you're doing long-distance traveling or having to get up at the wee hours of the morning or something like that? Exciting times. Speaking of sleep, I have it on good authority that we're going to be having another sleep-based episode in the not terribly distant future. Rhiannon seems to have found a really interesting expert for that subject matter. I'm always fascinated. I think we're all fascinated with the things we don't have quite enough of in our lives, and that definitely holds true for me in sleep. So be looking forward to that episode coming up relatively soon. Also in the pipeline, we've got a racetam episode coming down the tracks toward us, and another Know Your Neurotransmitters episode for dopamine. Not exactly sure what order those are all going to land in, but just a little preview of things to come on Smart Drug Smarts. And in the listener interaction department, happy to say picked up 2 more 5-star reviews on iTunes since last week. This brings us up to 93 on our road to 100 reviews. I mentioned the last 10 to get us up to the 100 mark. I was going to put in some sort of random drawing for a fantastic prize to be named later. I will stick to that. So now we have 2 more contestants for that, BD Wheezy Face and Fun Guy 327. Wheezy Face said, great sound quality, great editing, great topics. This podcast is a diamond in the rough. And Fun Guy 327 went on at greater length, saying, among other things, that Jesse puts a generous helping of quality into his work. Each episode revolves around a subject that winningly resonates, even if it's not about smart drugs per se. And most often, Jesse brings in the expertise of a professional in the specific area of discussion, guiding them to delve into the nitty-gritty details. So thanks so very much to both of you for taking the time to navigate your way through the iTunes interface and leave those reviews. Definitely really helps in getting the word out there. And I'm just excited to soon be cracking triple digits on those. But as Fun Guy mentioned, it's all about the experts on this show. So let's bring in our experts for this week's main interview.
Smart Drug Smarts.
So during one of the outtakes of the interview for episode number 71, Professor Scully mentioned something that some of his colleagues were doing relating to understanding the mechanism of action for fish oil, and that essentially what might be going on that makes fish oil a beneficial supplement for the human brain is that it doesn't necessarily make you smarter, but it allows your brain to work less hard in doing the same amount of cognitive work, which almost is like another side of the same coin. And my ears perked up at that, and I said, ooh, that sounds like an interview subject right there. Could I get introduced to your colleagues who are involved in that study? And that's what led to the conversation that you're about to hear. This is with Dr. Andrew Pipingas and Professor David Crewther, both from the Swinburne University of Technology. Dr. Pipingas is a researcher in cognitive neurosciences with a focus on brain electrical activity mapping, computerized cognitive testing, and biochemical markers, and his interests focus on the investigation of the effects of nutraceutical and pharmaceutical interventions on cognition and in the prevention of cognitive decline. As for Professor David Crewther, he began his career as a theoretical physicist, and his interests have since moved into neurophysiology, and he does a huge amount of work relating to vision, including neuromechanisms of refractive control, neuroscience of normal and abnormal visual development, and more. His studies have implications in dyslexia, autism, myopia, ADHD, and the understanding of conscious awareness and mind-brain relations. So we start off talking about this idea of neural efficiency and does omega-3 supplementation allow our brains to do the same amount of thinking with less effort? The first speaker that you hear will be Professor David Crewther.
Well, I think it requires that we define the ideas of neural efficiency. When people start talking about improvements in cognitive function by taking supplements of any sort, or pharmaceuticals for that matter, one has to know how you're going to measure that. And one side of the neuroscience of this, we've been developing techniques that really are targeting understanding efficiency in a sense. But the ideas of neural efficiency go back a long way further, probably to Richard Heyer in 1988. He started doing some PET studies looking at glucose metabolism in people with different levels of IQ. And to put it in a nutshell, if you like, the idea of neural efficiency is that smart brains work less hard. He followed that up with a couple more papers using Tetris tasks and so on. And then a couple of functional MRI papers using brain scanning came along, dealing with child groups with mental retardation versus controls. And to give you an idea or an example of this, paper by Quan and Rees was looking at a working memory task, which is a central cognitive ability, where they are asking children to do sort of a one-back task where they're looking for a repeat of something in a sequence of items that comes up. And the children with mental retardation actually activated at a higher level than the controls for this rather easy task. But when the task was made harder, the children with Fragile X mental retardation, the activity actually dropped away and the activity in the control group increased. So you get this idea of sort of a negative correlation between IQ and brain activation in the sense when they have to do particular sorts of tasks. And so some of our work has been aimed directly at trying to understand that. So the review really came a bit after we'd started all these investigations, and then we started to summate what we've been doing.
Tell me what prompted this study to begin with. Why did you start this review of the literature?
Well, perhaps I'll hand across to Andrew Pipingas because he's got a long track record in nutraceutical clinical trials and so on.
Yes, thanks, David. Well, my interest in omega-3s, as David mentioned, is because I'm interested in the evidence base for nutraceuticals and for nutritional supplements. When we first started this research probably 10, 15 years ago, there were very few studies, proper sort of randomized clinical trials in this area. And with respect to omega-3s, I guess I've increased my interest over time, particularly applying our clinical trials methodology to looking at what are the cognitive effects of omega-3 supplements, in particular EPA, DHA, fish oil supplements, and what some of the mechanisms by which omega-3s influence brain function, both cognitive function and more sort of neurocognitive function using some sort of neuroimaging and EEG type techniques. The literature was very mixed in terms of clinical trials. There was a very, very strong epidemiological population-based literature coming from cardiovascular area, looking at, for example, those with a higher level of long-chain omega-3s, EPA and DHA in their system also had a lower incidence of cardiovascular disease. There's also been a lot of literature on the Inuits, Alaskan regions and beyond. I think overall these populations have sort of high-fat diets and high tobacco. And this really interesting research which shows that why have they got such a very low incidence of cardiovascular disease? And it's been associated with their high consumption of fish, well, in particular seal blubber, if you like. So, those really oily fish is, what we're talking about here in terms of marine sources of omega-3s, and that's been the main reason for the lower incidence of cardiovascular disease.
There's this omega-3, omega-6 balancing act that we all need to do within our diets, and the traditional Western diet these days tends to be completely overbalanced with omega-6s. And so whenever you talk with people that are into nutrition or into the study of physiology and science, it's always kind of omega-3, omega-3, omega-3. You're not getting enough. There should be more supplementation. Is there any particular reason why one should be cheerleading for omega-6s? Is there similar downsides to having low omega-6 in that ratio?
Look, it is a balancing act, as you say. Omega-6 are important. They're polyunsaturated fatty acids. They make up blood vessel walls. They're important for all sorts of functions in the body. What we're saying here is that they're, as you say, in a Western society, they're taken in excess. The omega-6 equation, if you like, the conversions of the short chain to long chain omega-6s tend to produce more pro-inflammatory prostaglandins, whereas the omega-3s, the prostaglandins there, tend to be more anti-inflammatory. So you can imagine that resulting in this balance of inflammation versus anti-inflammation. The excess of omega-6s may be causing some harm in the body. And of course, omega-6s feature in a lot of processed foods and in vegetable oils. So yes, taking more omega-3s is important, but also taking less omega-6s, but still needing to take omega-6s in your body. If you're summarising the literature, bodies all around the world, the Heart Foundation in Australia and in the US and so on, are making these recommendations for at least 500 milligrams of EPA and DHA per day in our diets. And that's important for cardiovascular functioning and health. And I would actually suggest that the way the literature is heading, that perhaps, we should be taking even more than that, and perhaps the more vulnerable groups as well.
Tell us about the consistency of what you found when you were doing this review of the literature over the past 15 years or so. Were there any outliers or counterbalancing studies that seemed to go in a different direction? Was it pretty consistent?
I think you'll find that in this area, while, as Andrew says, the level of evidence for cardiovascular benefit is absolutely clear, the level of consistency of finding for cognitive studies is rather less. And that's partly because of the nature of all of these studies, partly because of the complexity of the brain.
Can I just jump in there as well? And with respect to cognition, I guess there's two broad areas we're talking about here. There's the literature on cognitive effects with fish oils, and what we've been doing more recently, what a lot of people are doing more recently, is using neuroimaging techniques, looking at blood flow in the brain, looking at the electrophysiology, looking at fMRI and so on. In terms of cognition, there was mixed literature. Some studies finding positive effects for cognition, a lot of studies not finding much at all. And more recently, it's been, I guess, summarised in a nice paper in Nutrients published recently by Clemens Bonshaki, where he talks about the problems with the methodologies. One really important aspect of eating fish or taking fish oils is there's a huge variation in the population in the levels in the body. So you can imagine if you're looking at a clinical trial where you're comparing a group taking fish oil and a group not taking fish oil, where they start off at baseline is very different. That variability, particularly in smaller numbers, in trials with smaller numbers, is one of the main reasons, I think, for some of the negative findings.
I've got a question about the digestive physiology of omega-3s and omega-6s. If we eat something like protein, our digestive tract breaks it down into amino acids before it's reutilized by the body and built into all new proteins. Omega-3s and omega-6s that we take in through our diet, do they basically remain omega-3s and omega-6s when they're put to use within our body, or are they precursors to something else?
That's a very good question. Look, I'm not a biochemist, but my understanding is that they're carried into the cell membranes as they are, but there's different transporters. You know, the EPAs and the DHAs, they can be bound to triglycerides, they can be bound to phospholipids, and this is the various forms of omega-3s. You've obviously heard of krill oil and fish oil. There's been research to show different bioavailability. But from my understanding, we know that DHA makes up about 30% of fatty acids in the brain. And in the retina, of course, there's very high concentration of DHA.
And they come under the name of essential fatty acids. So from the point of view, you tend to produce rather less of it than you need. So generally speaking, our consumption of DHA and EPA is highly necessary. The levels of EPA in the brain are rather less than DHA because there seems to be conversions between some of the fatty acids. But one of the interesting things that seems to have come out of some of our work is that it appears like EPA, eicosapentaenoic acid, may have some effects that are rather more rapid. We find some effects that are quite acute in terms of behavioural and also physiological change. However, we get measurably and consistent differences for EPA at shorter times than that, and it raises the question of whether essentially free EPA in blood plasma and so on is having effects.
Yeah, more acutely. So a lot of the research is around more the chronic effects, looking at that model of incorporation into red cells and tissues. And red cells is a surrogate measure of incorporation into tissues. Do EPA and DHA work acutely? Can they be taken up in the body much more rapidly, just like some of the other smart drugs that have been discussed on your show? And, of course, the vascular system is very intimately tied into the brain, so it's not surprising if we are getting commensurate improvements in cognition. That study hasn't been done as yet, those acute effects. I haven't seen anything to look at the effects of fish oils on the brain acutely.
So back to the principle of neural efficiency that we talked about earlier, all things being equal, if you have the same amount of glucose in your blood and blood flow to the brain, that a smarter person is going to be able to do more thinking with those resources?
That was really the outcome of Hayer's original thought, Richard Hayer's initial thoughts on neural efficiency. And that, of course, was between individuals. The interesting thing we did here was to actually go within individual, so to say, can we apply the principle of neural efficiency to an individual and say, what is the effect on neural efficiency within the one person under conditions, let's say, of administration of certain polyunsaturated fatty acids? And it's an interesting extension because it's also, for purposes of, say, brain imaging type things, it allows you're dealing with the same brain, so your chances of developing a better picture are much better. Getting back to your asking about this question about this neural efficiency aspect, I think you have to play into that the fact of studies where people have loaded cognitive demand more and more. And so, for example, say a working memory study, which you can make harder and harder, and ask the question, what happens in an individual? And the answer is the working memory network, parieto, dorsolateral, prefrontal, anterior cingulate cortex network, which comes up in every single study on this, shows increased activation when you make tasks harder until you get to a point where it gets too difficult and you get this sort of despair notion and people actually stop doing the task.
Also, the other interesting aspect of all of this is when you're comparing behavioural results with the fMRI results, quite often what you see behaviourally is very similar performance between participants, between subjects in these studies. When you start to look into the brain in terms of the activation through fMRI or other methods, you can see a difference between the levels of activation in the brain. I guess what I'm saying there is just be careful from behavioural endpoints that they may not be telling the whole story.
That raises the question of if two people are having the same behavioral endpoint, so they've both figured out the answer to a hard question, but one person's brain is cruising along, barely burning any glucose, and the other one's huffing and puffing and operating at the extent of its capabilities, does that really mean that there's much of a deficit for the person whose brain is working harder?
In the following sense, you take the performance in exams. If you work hard enough to get an A, then you've done all that's necessary to succeed, if you know what I mean. But yes, you take groups of people, individual students who are getting A's, and you'll say some are working much, much harder than others. Now, the real question is there I think, some sense of downside, if you like, to continually driving your brain to work hard when it's not that capable?
What are we talking about absolute numbers here as far as percentage of extra blood flow or extra energy expended by the brain for the people that have optimal levels of omega-3s versus those who might be lacking?
In terms of cognitive effects, not so much effort, but if we're looking at the sort of differences that we're measuring, say with nutraceutical effects with cognitive tasks, have been equated. If you look at measures of fluid intelligence, whether you're looking at spatial working memory, episodic memory, attentional changes across the lifespan, you can somehow calibrate that, if you like, that curve. Those are quite often linear changes. And if you use a graph like that to equate changes in, say, the response time to a task, you can work out a measure of years recovered in terms of sort of an anti-aging effect, if you like. And changes typically around 60 to 70 milliseconds, which don't sound like a lot. In the context of aging, that could quite often be equivalent to 5, 6, 7 years of years recovered in terms of the anti-aging effect.
One thing I'd throw in there is the question of stress. So while it probably doesn't do anything bad cognitively to keep performing at a high level, there are all these strange measures that probably do relate. But I think stress and the effect of continued stress as one gets older, that might be an interesting question. I don't think there's a real answer.
So based on your studies and understanding, what do you feel is the ideal dietary ratio of omega-3s and omega-6s that people should try to aim for if they're willing to pay attention to such things?
Okay, look, I mentioned before that there's recommendations by the Heart Foundation of taking in at least 500 milligrams of EPA and DHA per day. In terms of the ratio between EPA and DHA, the jury's still out on that one there. The so-called omega-3 index, which is a combination of EPA and DHA, I think that's probably the most important. Just getting both of those into the bloodstream, into the tissues, is what's important here. So I would talk about those together, and I would say, the recommendations are at least 500 milligrams per day. But to also eat fish each week. I think the issue around eating too much fish and getting it through just dietary forms is there's sort of the safety around mercury and toxicity there. So while the Heart Foundation is making recommendations that you should eat fish every week, that you should also include fish oil as a supplement. I guess some of the way of the future in terms of omega-3, looking at omega-3 research, we've touched on this through this discussion, is new methods going forward, which are neuroimaging, MRI, looking at MRS spectroscopy to look at neurochemical changes. Getting a handle of the mechanisms is really what's exciting about the future here. So being able to look at the impact of EPA and DHA on the brain and to associate it in terms of cognitive functioning with things like BDNF, the issue of neuroplasticity, looking at the effect on blood flow in the brain. This is where we're at now. A lot of studies starting up.
From my point of view, I think getting into the neural efficiency questions again and trying to take them through further. If you consider the simple idea that smart brains work easier, it's very hard to actually then go to a brain and say, well, which part of my brain is working smarter, because there are so many bits and pieces. And I think the approach that has to go forward is to understand the networks, so the parieto-frontal network of attention and its role in performance here, its role in intelligence, and so on needs to be better understood. And then within that framework, to then go back into the questions of neural efficiency to ask the proper questions. In neuroimaging studies, you'll find bits of the brain that increase in activation, bits that decrease in activation. You'll find increase in the percent bold response. You'll find extension of the areas that are activated and so on. That stuff needs to be sort of improved so that we can get a better measure, if you like, of the function of networks, the role of these various networks in understanding intelligence. And I think intelligence itself is something that is a worthy target of applying our ideas to.
Smart Drug Smarts.
So thank you so very much to Dr. Pipingas and Professor Crewther for taking the time for that interview. I think that's our third fish-related Smart Drug Smarts episode, probably not to be the last. Like I've said before, doing this podcast has legitimately changed my diet quite a bit. I'm eating significantly more fish than I did at the start of this. And I think it was episode number 41 with Dr. Michael Lewis was the first time that we talked about fish oil. Some of the dosage numbers that Dr. Pipingas just mentioned in this past episode were significantly less than what we'd heard in some previous episodes. But as you mentioned, all these things should sort of be balanced off against how much omega-3s is coming as your normal food diet, whether further supplementation might be necessary. There's also omega-3s in non-animal sources like avocados and nuts, although there's a certain amount of greater bioavailability from animal-based omega-3s than plant-based omega-3s, as I understand it. They're more likely to actually cross the barrier of your gut and wind up in your bloodstream. So as with all these things, you can go pretty deep down the nutritional rabbit hole in trying to find out what's best. But the conventional wisdom is if your diet is anything like the standard Western diet, chances are you're probably tipped too heavily on the omega-6 side of the omega-3, omega-6 fatty acid balancing act, as omega-6s are hyper-prevalent in most of the foods that we eat, whereas omega-3s are really, unfortunately, fairly underrepresented. I thought it was a really interesting point that Dr. Crewther made, that two people might be able to get to the same cognitive endpoint as far as concentrating on a problem, thinking through the answer, and getting a correct result, but that the person whose brain is working less hard, in this case because they've been able to get that correct macronutrient mix in their diet, is essentially having less wear and tear on their brain as it goes through its thinking processes. So while something like omega-3 supplementation might not make you overtly smarter this year or next year, it might be the kind of thing that over the course of a lifetime, your brain ages less because there's less hard work of use in the intervening time, and you might be a 70-year-old with a brain of a 55-year-old as opposed to a 70-year-old with the brain of an 80-year-old if you made some different lifestyle choices. But that's all the fish talk for this week. Let's do the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
So everybody's interested in sort of weird paranormal phenomena. How can you not be? I remember when I was a little kid, I had this convinced belief that I could tell when people were looking at me without actually knowing they were looking at me. Like you could feel their eyes on you. And at the time when I was a little kid, I really felt that was true. Anyway, it was just an example of my own life kind of feeling like, well, wait a minute, that's not like one of the 5 senses that they have listed in the textbooks, but it felt like a sense that I possessed at the time. And anyway, scientists are studying this other sense, which seemingly exists, which is our ability without looking at things, or at least without consciously looking at things, to have a pretty good idea of some of the physical dimensions of the stuff that's in our immediate area. So what was happening? Scientists in this study, they had subjects focus visually on some particular thing, which wasn't what they were studying, but they would be looking straight at one thing, and then they'd have a bunch of other objects in sort of their peripheral vision that they couldn't really see clearly, definitely is not in the part of their vision that is being paid attention to, but the blurry sides of your vision. And they would have a table in front of them filled with different objects at different distances, so you couldn't really judge the size, closeness of these things because they're off in the periphery of the vision, but they would have the person, nevertheless, while not looking at these things, reach to pick them up and hold their thumb and forefinger the right distance apart to grasp the object, or just to estimate the object, not even reaching to get it. And humans, it turns out, have the uncanny ability to correctly gauge the size of things, despite all these visually distracting cues and the fact that they can't be looking at them directly. The research team found that even when participants had no idea what the size of the objects were, their fingers opened just the right amount to grasp the object without fumbling or dropping it. This finding is quite remarkable, explained Juan Chen, the lead author of the study. It shows that people have the ability to perform actions towards objects even when the key feature controlling those actions is not accessible to the brain. We propose that there are 2 separate but interacting visual systems, one for controlling skilled actions and another for conscious perception. This work provides new support for that theory and sheds new light on the division of labor in the human visual system.
Smart Drug Smarts, the podcast so smart we have smart in our title. Twice.
Okay, that is the entire episode number 73. Thank you for hanging around until the end. If you're still here, if you feel like evangelizing Smart Drug Smarts to anybody around you, we heartily encourage you to do so. If you're sitting in class, you have an extra piece of paper you're looking to get rid of, why not write smartdrugsmarts.com on that paper, turn it into a paper airplane before you huck it, and you'll be spreading the word. The show notes for this episode will be online at smartdrugsmarts.com as usual, including the links to everything that we talked about here. I'll be back at you next Friday, same time, same podcast, and the same unflagging commitment helping you fine-tune the performance of your own brain. Have a great weekend, correctly manage your circadian rhythms, have some fish oil, and good luck in grasping for nearby objects. And of course, stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only. Although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.